So, just to point out what's happening here, um, I do have this in Spanish, mi unidad, my unit, uh, collab notebooks, and it uploaded um, the image processing notebook from today. If you can do that as well, will be nice. I will review what we did the last class as well. And also I uploaded the set stacks here, which is the control here, 40x, and the wild type arsenite, which is the puncta containing set stack. Okay. So if you can upload the notebook, you will see something like this. Um, Basically, I just run this, uh, the library requirements. Um, I run it before in advance, so basically it's just telling me that all the requirements are already satisfied here. So we're good to go, I guess, uh, from the Jupyter Notebook. Um, Please let me know if you have any single problem running on Colab or the Jupyter Notebook, or if you created a new environment. Okay. You can see a nice log here, which is the progress bar. It's a green triangle. You can see when the triangle moves in which library is working right now. Okay. So the green tick means it's universally that it means with actually the installation ended. So you will see the log file here. All the requirements are already satisfied. In this, by doing this, we overcome all the problems we had in the past class. Okay, so we can load the libraries here. You see the run button here, which is execute cell, and in Spanish ejecutar celda. So we'll click on that, and basically in 0 seconds you can import all the libraries you require. Okay, important thing here is that um, basically you have the AI CSI image IO, which is library um, important for loading images. Could be a set stack from Carl Zeiss, or could be set stack from other microscopes as well, or could be just a TIFF, a stacked TIFF image. Okay, so if you have problems with compatibility or you don't have a confocal microscope from the Carl Zeiss company, please export that to as a set stack TIFF format, and you are good to go. Okay. Um, yeah, we will see what's going on here. Oh, you have to, in this case, you have to load the content in the folder content, this this file. So I will double check where is that. Or maybe I can change here. Oh yeah, I need to upload it in the content file. So I will try to figure out that now. But I will switch to my local Jupyter notebook as well in the meantime. So what I usually do, just to refresh what happened the last time, um, say Jupyter. When you call Jupyter Notebook, you will see something like this. Okay. So in my case, I have everything linked to OneDrive here. And in this case, I will open here desktop. I will go to UCC, and then I will go to my folder here. I will surf into my folder, uh, just as I did last time. I have my note here. I have everything here. If you are working on Jupyter Notebook, I do not have to run this install again. If you run it just once, it's okay, without warnings or errors. With warnings, but without errors. Okay, so I would just go straight by doing the importing the libraries. So I'm expecting to have a log here, uh, which is it's okay. So basically, just a single warning here. Um, can you tell me, maybe more people is joining right now, but uh, do you have any problem running this cell? Can you put in the chat for me if someone has a problem, single problem running these lines? No problem. Thank you. Thank you. Awesome. So I just will wait maybe for hello to the people who are actually connecting. Hi, Tyler. Uh, Ian, hi. Uh, yes, just reviewing two methods to run these notebooks. Not very familiar with Colab, but a lot of people did, and it worked out. So I'm very happy for that. Colab is here. What does the show the screen here? Here. So basically, the all when you run the installation, all the requirements are already satisfied, and in a couple of minutes. So the only thing you have to specify the path here. If you have a specific folder on Drive, you have to put it and you will read the files. I prefer to do in Jupyter Notebook, but if you won't go for Collab, that's okay. That's totally fine. It's actually even better. You do not waste CPU requirements on your computer. So you have these nice animals, cats and dogs, who actually has the Halloween theme, because Halloween is approaching. So uh, it's very nice. But I will switch to Jupyter Notebook right now. Here. And I will we loaded the first sample, which is the control. Usually we do a control in microscopy here in order to set the background of the fluorescence. That's very important because sometimes when you have problems setting the threshold of what is background or not background, it could be tricky within experiment to experiment. So I will open the control file, which it says here control. I just put a message here, Stack Overflow, ChatGPT, etc., etc. Please explain this code, and you paste the code. If you put in ChatGPT or Stack Overflow, which is in Google, Bard, you can use Bard as well. I don't know about Perplexity, but if you put please explain this code in plain words and you paste the code, you will have an explanation for that. So just a reminder, we need to use these tools wisely. So it's a good help. Um, Basically what we did here, we parsed the metadata. So this is a text file, this set stack. So basically the root has a specific, you can call, in order to call the root, you have a specific function from the libraries we loaded to read the metadata just as it is. And you can find all the channels with find all function from Python, all the channels that you recorded, maybe green channel, red channel, DAPI or Hoch in maybe in German, uh, to see uh, to see the blue channel as well. And you will create an index here, an indices. and then you can print the channels as well. How many channels do we have here? Since it's text processing, we have duplicated names, but we have three channels here. Okay, uh, and this library, which is very nice, AICSI, AICS, Image IO. You import this library, which is the IICS image. And from the writers, you import one TIFF writer. I will put a comment here, and I will send this Jupyter notebook for you guys. But if you want to take notes on the Jupyter notebook, that's very helpful for the future. With this function, you can write images. For instance, if you had to want to paste image in PDF format or in TIFF, in this case, this is specific for TIFF format. Um, you can interconvert formats. This is your library, and then you have everything uniform in terms of the images without losing resolution. We will load the image here, this directly from the set stack. And usually, when you take the picture, you have the image size in microns, uh, but you can easily find that with 5G. It is just image J. That's what they claim. They claim it is just image J, but 3G or image J, they will say the dimensions of the image. Okay, so we do the pixel dimensions, and we will calculate the size of each uh, pixel by dividing the image size by the number of pixels. Usually that's all the information that it will give you image J. How many pixels will we have? Usually it's 1024, but could be less or more, okay? Depending on the size of the image you took.
Carlos,
Yes?
how did you get the pixel dimensions in the data?
By loading the set stack directly to image J or PG, PG. I don't know how to pronounce
Okay,
that
so
one.
you got it
Um,
from Fiji.
I will explain that right now. So
Okay,
I will
thanks.
do it. Yeah. So that's a good question. Usually you PG, but maybe you can, as Stefan pointed out last uh, class, you can actually figure it out with Python itself. Uh, but I am for sake of not to do more coding, I would just show this one. Okay. Just give me 1 minute to surf into my folders. Yes, do we have this one? Okay, so we'll try to. And you see my screen, um, Felipe, are you there?
Yes, I can see.
Okay, so you see here 354.25 square microns, right? And you see
Mm-hmm.
another number here. It's not brackets, but 1024 square. So this is the pixels.
Okay, yeah,
And the format
thanks. Yeah.
is 16 bits, could be 32 bits or 254 RGB, depending what you are doing here. And 54 megabytes. That's the weight of the file, the size of the file. But usually the first two measurements are important, microns and the pixels. Yeah, good. Thanks for the question. Uh, and now we'll Share my screen. Okay. So basically, what we're seeing right now, it's um, the pixel size, obviously, according to the image. Okay. And then we will write with this one TIFF writer, um, the channel one and channel two. So I will also show what's going on here. If you go to 5G or 3G, I will just load one of the channels here. Okay, so I just load the channel one, and you can see here a nice stack of the nucleus staining, DAPI in this case. Okay, can you see that? Or yeah. Okay, so basically, obviously, the other image is in. Great, great, good. So basically, the other image contains the puncta in this case. So I will load that as well. Okay. So we have the channel two here. Um, this is the control, I guess. Control, no, sorry. We'll load the other one. Yes, this one. This is the other channel. You will see some puncta here, which are stress granules caused by arsenide treatment on the cells. So you will see the other channel here, right? And the aim with this specific experiment is to count the number of puncta per cell. Okay, so we will go back to the Jupyter notebook here, uh, and we will do a reconstruction we did last time. So we will load the libraries here. We will load the images we actually wrote, which are in TIFF format here, which is the first channel and the second channel here. And we'll do some tricks here. Sometimes the signal is super low compared to that. In order to have like a nice ensemble, you can enhance the signal, but that this is just for training purposes in terms of the algorithms or for counting content. Actually, you have to always present the raw data and how it looks. But for purposes of training on or running the algorithms, you can do these tweaks with the data, and it's totally fine. It's just for training purposes, okay? And actually, the machine learning and the deep learning algorithms do that all the time. So we will increase, we will leave it as that. We will multiply by one, which is the same to just leave the channel at is. But if we put two or even 10, for instance, you will increase the signal. It's the same what the confocal microscope do when you ask or when you try to increase the signal because you are not seeing so much. Okay. Um, and we will display the maximum intensity projection, which is it's an ensemble of the pixels here, and you will try to recreate the maximum signal per position here on the image. We will display also the nuclear staining here, which is the dapping here in blue. Usually, you can change this of color. For instance, you put greens here, it will appear in green. If you put reds, you will see reds. And actually, I will tell you what the colors. Um, yeah, sometimes it gets stuck. So, can you see this? My screen, the Matplotlib. Um, if you can confirm that, will be nice. For you to show how many, the whole palette of colors. Thank you, Adolfo. What you can do with the Jupyter Notebook, and you can put everything here in order to, for instance, if you have multiple channels and you want to do an ensemble of the data, or do an ensemble of the images, this is your friend. Matplotlib has a lot of colors, and it's very nice. So the whole names you can find it here. Yes, I add the reds and blues and greens. This is by default, uh, because sometimes the Seaborn, which is the other one. I will put this link here into the Jupyter Notebook. Here. Greens, blues, and which are called the color maps. This is the other one. So this is the other uh, uh, regex you can put here. You can use the yellow or red, purple, red, or green, blue. It's usually very useful for the heat maps as well. If you do heat maps, usually do these colors as well. So I will put these two here, and I will change the cell here to markdown. So I will have the links here, and I will obviously go to the link when I clicked. I will click the other one. No, the first one, sorry. Here. Um, but also, what I will do, I will insert a cell above with the A, capital A. I will put 4 hashes, and I will put hash, 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 um, color links, color palette. Okay. So, yeah, for your reference. Oh, can you send me Stefan? Can you send me the error you had, like in the chat or for everyone? Oh, when you change the color, that happened? Oh, interesting. So, did you try it on your Jupyter Notebook or in Colab? A notebook. Okay. Maybe it's a good idea to try and Colab as well at the same time. But send me the error. If you can uh, copy the last part, the last bit of the error, uh, as much as you can, not the whole thing, but the last part would be helpful for me to see what's going on. deflate or clip deflates set leaf the compress. Do you load the images not the uh, because this was compressed in zip format. Did you decompress the whole directory and you have nothing compressed, or do you have? Okay. Can you try on Colab I don't know if you have a folder. You can upload the Jupyter Notebook with the data within the same folder and run everything from scratch. I mean the new notebook, the notebook I just sent. If we can upload that with the two set stacks, will be nice. Maybe it's something that you missed since it's a new notebook. This set leaf, it's very important. Um, I will talk a little bit more on that. Uh, yeah, I will try to see something in English here. Fortunately, it's in Spanish. Sorry. Probably just disconnect. Sorry about that, guys. Um, yeah, I was talking about Satlib here. Um, so let's see. So this is the description of the library. It's a memory compression library. Is important for decompressing things. So if you have problems with that, you have to do a workaround. Okay, uh, on internet. But it's very important to have this library up and running if you have a new notebook. I don't know if Tyler, do you have problems with Setlib at the beginning since you already bought a new notebook? If you can put it in the chat if you have the problems with that. Sometimes it comes uh, with the notebook, or sometimes it doesn't. So okay, no problem. Okay, yeah. Okay, so next thing we did a blend here in order to have the common picture. You can obviously achieve that with the ImageJ at ease here, um, which is code block. You will see uh, the blue and the red merge or blended. So, and you will also write the overlap here in PDF format. You can write it in TIFF format as well. So this is the control. What happening with the Arsenite here? We open the file, same thing. We parse the metadata, store the channels, read the image with IX image. Um, the pixel dimensions are the same here because the image was taken the same day in the same microscope, same settings, the very same settings. We also load the same libraries for sanity check, uh, and then we will load the set stacks, And so we have puncta here, obviously, because it was treated with arsenite. It's a very toxic metal, heavy metal. So basically, will the cell will react to that. Uh, and then we have the puncta here. I don't know if you can see it well, but maybe in your own Jupyter. Uh, and then brings to the nuclei puncta detection. Okay, so we tried it last time. So we will import this sky image, which is the library I talked last time. Maybe I will refresh that. Scikit image, right? The Scikit image processing in Python. I will put the link here on the Jupyter notebook here, uh, right, for you to have it. Basically, we will do the same, but in this case, we will load not the set stack, but the TIFF stack, right? With the channels, the convoluted, okay? So the individual channels, channel one, channel two. And then we will do some adjustment for 3D images, which is called from the NP. It's equals the NumPy library. We saw that with Sebastian because I will just remember that this is part of the bigger course on data science, and um, I will encourage you to read the basic libraries in Python, which is NumPy. Just take a look. PD, which is pandas, what's going on with these libraries. But the NumPy is the numerical library in Python, and pandas is for data frames manipulation. Okay, so we did try to process the puncta. So what's going on here? This is a code block which is kind of very wordy. Sorry about that, but I will try to break it down from step by step. So we will load the set stacks with a progress bar. So one funny thing of Python is the TQDM. So adds a progress bar when processing things. Okay, so it's very cool to have the TQDM library if you do image processing on Python. So because you will have sense what's going on if your analysis will take 1 week or a couple of minutes. Okay, big difference. So I like to have that. And then we will subtract the background. Obviously, we have the red MIP control normalized, which comes from here. So this is tricky experiment because it's not the background here is not just a black screen. It actually is signal. It is fluorescent signal, but the protein is evenly distributed along the cell. But with arsenite, something very interesting happened. That punta is started to appear, which is the stress granules. Okay, in Spanish, gránulos de estrés. So basically, these stress granules. Um, uh, it is a way of the cell to respond to a specific stress signal by stalling translation, for instance, and other processes as well in order to survive. Okay, so basically, this is not just we're using like like the black background here. Actually, the cell the protein is aggregated, and the fluorescence is you can really distinguish by eye the puncta versus the background here. Okay, so that's why we loaded this red beep control normalized. We will clip the values to ensure no negative values as a subtraction. This is, in other words, we do not care about the black background here. Actually, we care about the red background, which is the evenly distributed signal, not the puncta. We will use Laplacian of Gaussian to the blobs, so which is the puncta. Um, it's an algorithm, very old but very useful. That actually it's a mathematical transformation of in a 3D space. We usually do integrals or to integrate with certain numbers. For instance, if we give random numbers, we will calculate the Laplacian, which is a mathematical transformation, and then the Gaussian. The Gaussian first, and then we will calculate the Laplacian of the Gaussian. If you are more interested in that, please uh, try to figure out that with a proper person, which could be a mathematician. I just use this thing, but it is very straightforward um, thing to detect puncta from uh, unevenly distributed space to detect deviation or deformation in the space within a 3D space. So you can see parameterization here, which is number sigma, max sigma, and you need to play with that sometimes. I would say not all the times, but sometimes you have to play with these numbers in order to have best results. Um, this is why I do not like so much of these methods because you have to parameterize. We'll write down because I don't know how to pronounce that well. But that is- So this is a problem in image processing when you have to change a lot of parameters. Because it will you will not be able to generalize a method. That means your method works just for this data, not for every single data. So that's the aim of machine learning and deep learning. It's um, a method who actually, if you try to keep it consistent and as much consistent as you can try to generalize a specific method and try to process a lot of images uh, at once. Okay, so just give me 1 second. Uh, okay, so basically we will count the number of puncta and we will do a visualization, which is matplotlib here. Matplotlib library it is usually for, maybe the problem is a damage in the archive. Try to download again. Okay, Adan says that to Stefan. Maybe it's that. Okay. And then we will try to run this thing. We have the progress bar here, which is the TQDM library, which I like it. We'll try to detect the puncta here. Um, so this is a slide two, slice, sorry. So you can see here in the progress, the more and more stress granules are detected. Some big ones, as you can see here, I will try to, oh, this is, it should be different color. Uh, just put big punta. Here, it's a rounded circle. You have another one down there, but sometimes it's different. It's not just a single dot. Sometimes the punta is very big. So this is a good method to distinguish that, okay? Okay. Um, so we finish at the end. Total red punta across all slices are 629. But since this is the set stack is not so deep, it's not like in this case it's just cells. If you are working with with a thicker tissue, sometimes this is a good explanation. Basically, you ask. I usually go to Herban at CMA, CMA, which is a facility here of microscopy. Say, hey, I have a thicker fish tissue. Can you do a set stack, but jumping across the whole tissue in order to capture as many features as we can? Or sometimes I just need to quantify fluorescence and I just need do the very dense set stack with these cells. Um, because these are cells, we can collapse positions. But if you are working with tissues, just from, for instance, if you want to quantify fluorescence signal from vibraton, it's called. Correct me if I'm wrong. Or cryostat, usually you don't need to collapse dots because the dots are contained in one set stack. I don't know if I explain myself well or probably not. But if you want to share experience with that, doing imaging on Vibratom or Cryostat versus cell immunocytochemistry, which is called, please let me know. But usually I found that the dots you can find, because the dots can be in the same position, but it could be two different dots. The one is in the upper part of the tissue or the lower part, but it could be in the very same position. Stochastically, it's not common, but it could happen. Okay, so that's why I prefer not to collapse, uh, not to collapse positions. So we automated this algorithm to collapse positions here. So basically, we will process also all the slices. When it will take a while, not so much, I believe. So this is the color CMAP 9P spectral. So it's a very nice color. Try to actually we have a bigger computer. This will take just a couple of seconds. Uh, 361. We can change the number here. You can change here, change the color. If you just need red, put it in red and not a spectral, which take us the whole spectrum of colors of Matplotlib, it's fine. It is just for detecting. Sometimes the spectra goes darker or warmer colors with the lower signal and bright colors with the increased signal. So it's very nice to visualize this one. 361, and we have the red puncta across all slices at 600, the double, almost the double. So basically, what's happening here across the slices, we are counting the same dot more than once. So that's what's happening here. In my case, this is just a cell. It's very flat here. Basically, we just collapse the positions. Question, same results. I will put in the answer here, no, since this is immunocytochemistry. Immunocytochemistry. Okay. Um, so not collapse. One. Uh, at the last one, I'm not quite sure, but in Vibraton, definitely I will not do that. Okay. And the DAPI counting, which brings to the next to this class actually, what's going on with DAPI. So I would like to ask you guys if you have any single question regarding the previous code piece of code we are showing. I know I didn't explain everything, but this piece of code can be customized for your needs in order to load your stack, see what's going on. Okay. I would like to know if you have any questions with that.
Also, I have a question. Can you get a shape descriptors of the punctures? For example, if they are like bigger or smaller, or if they have like different shapes. Like, can you get that data from the pictures?
Yes, actually, it's one layer of complexity, one other layer of complexity, and you can get the different type of dots. I never did that before, but I know you can do that very easily. So I don't know, Francisco, if he's the expert of stress granules here. If you have any experience counting different types of dots, and or it's a biological um, feature depending on the cell, or sometimes you find both. You're mute. I think you're mute. I think you need to disconnect the.
Sorry.
Yeah.
Now you can hear me?
Yes.
Yes? Yes. Okay, so stress granules assembly, it's a biological process which is dynamically controlled. So the stress granules are assembled step by step from a nucleating protein called D3BP1. So you have bigger and smaller spheres. So these are three-dimensional, and they grow up and then they disappear also. So you will have different sizes.
Perfect. Thank you for explanation, Francisco. Yeah. So basically, you can find both. Very interesting question. The puncture detection runs for each slice apart, or it works in 3D. In this case, with detected the slice, but you can do a volume reconstruction in Python and count it afterwards. Okay, so you can do both. Um, okay. So basically, this is more tricky, and that brings us to a very known topic of deep learning and machine learning. I already know you guys have some experience with that. ChatGPT, you heard the robots that walking alone, taking decisions, a lot of things going on. Uh, but it's important for imaging processing because it's getting more and more attention in terms of processing big data, uh, substantial amount of images with computers. So it's very interesting what we can do with that. In this specific case, we will do just a custom, not a custom, just a standard um, nucleic processing with that comes by default on Python. Okay, and you will see it's very bad. So my question is how we can improve that. Okay, so we will load here the scikit-image library, which is here. Uh, and we will import different features from this library: color, morphology, measure, filters, and exposure. Okay, so which are features we usually see on images. The morphology is rounded, or the measure is in fluorescence quantity. Filters, how many filters do we have on the data? Is RGB colors or not? And exposure, etc., etc. And we will load. We'll put just a for a reminder, scikit-image, and we'll put the matplotlib here as an example. So we will load the set stack here, which is the one who actually contains the puncta, and then we will convert to grayscale to just get rid of the color and just count black or white. We will enhance the contrast. If this is a black box, I will encourage to say that. La caja negra, como se dice en español, in Spanish. Black box is something that actually do something, but we do not have much control of what's going on there. But it comes with scikit-image. And we will do some thresholding as the documentation pointed out. on the code that comes from the scikit image here, which is documentation. For instance, here I show you last time here, uh, but we have a lot of different documentations. So basically, it is somewhere here. Um, and we will do morphological operations, which is you can adjust this size as needed, but this also is a parameter. You have to just write data. And we will do a binary selection because we need to, in this case, the background it is usually black. So we'll try to distinguish or discriminate the nuclei, which could be, it will come in white versus the background, which could be very dark or just black. Okay. Um, you have to adjust the area threshold as needed depending on the cell. And this is also ugly for me. I don't like it very much because it's also a parameter. Just okay. And then visualize. This code doesn't change so much. If you notice on Matplotlib, I don't know if you guys had previous knowledge with that, with Matplotlib. I'm not an expert at all on Matplotlib. I just copy paste the codes for seeing visualization. Sometimes it's shorter codes, but the Seaborn, this is from Matplotlib, but Seaborn library is shorter. Okay. We will see this, and this is interesting. So we counted 30 cells in this slide. This is correct or not? What do you think, guys?
Oh,
No. Okay. Thank you, Sebastian, for the feedback. For the guys for the other one. It's a common problem, usually in histology. In histology, uh, when you have a tissue, for instance, the cells are not equally or even distributed on the slice. Plus, for instance, is the if for the tissue is under development, for the people who actually is studying development, you will see enlarged cells, soma from one side to another, um, radial glia touching one edge on the tissue and the other part touching the other side, the classic definition of radioglea, you will see a lot of stuff going on. Small cells, rounded cells, clumped cells in the terms of blood um, imaging, um, gem sustaining, for instance, or for Plasmodium falciparum, or different stuff like that. So basically, this is this algorithm is far away from uh, reality. We need to work on that. That brings us to, unfortunately, deep learning and machine learning. So I would like to do a poll first with you guys. Who here have been working with that before? Have some experience? Not should be on coding, but should be by clicking, imaging processing, or another program. Do you guys have experience on that? Previous experience beforehand, or not so much? Um, Not at all, Francisco says. If you can answer, everyone will be in deep learning, machine learning, none. I've used LabKit for image segmentation. That's very nice. Ian, can you talk a little bit more about that? How it works? Do you have a sense how many, if they use convolution neural networks and some sort of fancy algorithm to do that, or that use machine learning? Okay, okay, yeah, that's good. It's machine learning is precedes usually it precedes the deep learning analysis neural networks. Okay, that's very nice. Well, sometimes to count nuclei, it's very easy by hand. We can count it here by hand by doing click, 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 click. But what if we have like 1,000 set stacks, or we need to count a lot? So um, that's a problem. And sometimes it happens more than you think. Even with your own data, you might think you have a lot less, not so much data, but actually you have a lot. Believe me on that. Um, and it's usually good to benchmark different methods. So we'll see that. Obviously, we have here. I will try to draw here something. Um, Unfortunately, the colors very bad here. It's I can't change from black, but I will try to write instead. Okay, text here. Uh, Three cells. We have three cells, for instance, here. Also here. Um, Missing cells. So this is because uh, the color, the thresholding failed to detect these cells. Um, and we have here two cells at the lower part. Okay, so that's a problem. Probably we'll ask machine and deep learning what's going on here and what we can do by hand. Okay, Kid is pay. Do you have to pay for that one or not, Ian? Curious to see if you have to pay that or it's open source.
It's a Fiji plugin and it's free,
Oh, nice. Very
but
nice.
it's still relatively new.
Okay. Okay. Sounds good. We'll get into that. Maybe we can surf into the documentation, see what's going on there. Uh, but I will do a brief introduction to deep learning here for you to see. Is this right? Obviously not, right? Um, so we'll do an introduction to deep learning, but first things first. I will try to explain this image first. Sometimes I repeat it in I'm getting shorter on sayings here, but in my country it says very much more than 1,000 words, more than 1,000 words. So una imagen vale más que mil palabras. Well, we have that here. So we have the inputs here, and these rounded circles, these circles here, actually are neurons. But you can. This is the whole. It's a whole concept, but the whole idea here is to build a neural network. Uh, Sometimes it differs. Some neural networks works better for images, others for data. Pandas, for instance, connect very well with neural networks. But you can see here inputs of could be anything, could be data of numbers, pixels, number of pixels, a data frame, an image, which actually we are addressing here, and a lot of images you can input, and then you will give a weight, peso, and this weight will tell you if this image has a lot of information in terms of pixels or doesn't have any information at all. It is background, and then you will do a sum of every single weight here, and you will program a function, uh, and then you will need to do an activation function. I know this is a lot to take in, but I will briefly explain step by step, and then you will have an output. Okay. Uh, Personally, I prefer to explain this by programming. but then I will address any single question you guys have, okay? So I will do some programming first. I will show you what's going on with the basic and standard deep learning processing, which is very human intuitive, and then we will go from there, okay? Okay. We will load the TensorFlow. Obviously, we need to go to Tensor. Let me see. Load the web page. Sorry about that. It's in Spanish, but probably if you put TensorFlow on your Google Navigator, it will appear uh, in English. Okay. I will translate here. Prepare data. So this is important because you can do, for instance, a large data frames or a lot of images. You will create artificial intelligence models here easily with TensorFlow. Actually, there's a tool which is called Keras. I will write it down here. Keras, and Keras will guess the best neural network for your data. It can learn from your data, and it will propose a good neural network. So that's a very interesting tool. Okay, Keras, and we'll use that, and then we will implement models, and then we will draw conclusions. Okay, based on that. This is TensorFlow. So I will just put the web page on the notebook here. Okay. So then we will load very interesting data sets called MNIST. Does someone heard about that before? MNIST or Ham to or Ham 10,000? Maybe in research or papers. Okay. So if it's not the case, the MNIST database in this case never seen MNIST before. Okay. The MNIST database contains 70,000, 70,000 handwritten digits between 0 and 9. So 70,000 persons were asked, no, less than that, maybe 35,000, which is quite a lot, or 10,000, no, 10,000 persons or 9,000 persons, which is a lot, were asked to write from 0 to 9 handwritten very rapidly, very fast. And they sent a picture of that to someone, and they gather these 0 to 9 numbers um, in a database. So basically, it's a whole challenge because when you write down very rapidly with no time, you will make a mistake in writing numbers. A 7 could be a 1, and it happened to me at the laboratory that actually someone said saw this. This is number 4. What is this? No, this is number 6, but my 5 has a little tail at the end. So basically, it looks like a 6. But I said this is kind of a square. This is a corner. You write down the 5 here, but no, the other person said for me this is a 6 because it's a closed circle. So this is a problem, and the machine and deep learning try to address that with its algorithms. Actually, this is a benchmarking, and you can easily import MNIST automatically, okay, by running this one. Okay, we'll run this one again and this one. Probably you will see some seconds of downloading if you did it for the first time. Okay, And then you usually train data. So basically, the data, which is, for instance, 70,000, you usually divide the data in three parts. The trained data, you train the algorithm with some subset of the data, and then you will test the data to see if the algorithm match with the training data, and then you will do validation by eye, for instance. Um, uh, for instance, you can easily see that this is a 4, but the machine interprets that as a 1, okay? So we will try to train with as much as we can. Usually we will train with 60,000 images here in your computer, and then we'll try to do a test data with 1,000 images, 10,000 images, okay? So this is happen very rapidly. And this usually comes with the typical notebooks that actually process the MNIST data, which is our friend Matplotlib. So probably if we put 330, maybe it will change, or maybe it will give you an error. If it should be 36 in this case. Yeah, but you can change this number as well. I will leave it in 224 to 12. It goes 6 by 6. Maybe 6 by it goes 1, 2, 3, 4, 5, 6, 7, 10, 12 by 12. Yeah, but this is just a subset of 12 images by row, and you will see the classes. So class 5, this is a 5, okay? 0, very easy to see that one. Class 4, this is a 4, 1, 9, 2, class 1, 3, 1, 4. Notice that this 4 is very different from this one. Okay. And then you will have the 3 and the 5. This could be this looks pretty much like my 5. If you put if you close here, it will be a 6 from some people. For me, it's 5, still 5. The 6 here for me it's like this. Okay. Then you have class 3, 6, 1, 7. It could be 1 here. Not so much difference here. 2, etc., etc., etc. And then we need to pass this data from 2D from 1D, one dimension. So we have volume, which is 3D, which we have an image, which is 2 dimensions, and we have 1 dimension. What could be that one? Usually comes with a vector, mathematical expression is a vector, okay? And we have to do that and reshape the train and test data sets. So we are training with the 60,000, and we are testing with the 10, I believe, okay? You can increase or decrease this number, 65, 50,000. So you can do that as well. Maybe here. Okay. To train this model, we have to normalize the weights. In this case, we will just normalize by dividing the max value of each dataset. What is the meaning of that? In terms of in an NumPy array, um, because you have positions X and Y, and you have numbers that it will give you the intensity of the pixel. Obviously, the maximum values of each position will be retrieved here, and you will normalize the entire datasets with that. Okay, in order not to train, for instance, what if we draw very lightly a 6, and we are training with very bold numbers. Okay, So that's a problem. So basically, we try to make as even as much as we can. Obviously, the max value is 1 and the min value is 0 because this data is already normalized. That's a small trick. That's why we are working with this data, just to do an example what's going on here. And we will define the number of classes. Then this function len equals to length. Okay. And then usually we load the Keras library here to transform categorical. What is categorical? For instance, image 1 equals to 1, number 1. Image 2 equals to numbers 9, for instance. Image 3 equals to number 10, for instance. No, 8, for instance, and so on. Okay? We have our model definition here. Just explain the very first part. And then we will build the convolutional network. So okay. So basically, we will import two libraries from TensorFlow Keras. Keras is, as I mentioned before or previously, is part of TensorFlow. We will import sequential and dense. We will need to do an activation function, which is important to initialize the neural network. So what is dense? Dense is a layer of neurons. And in this case, we have 512 neurons classifying the images. How can I distinguish that? What if I put 100, 128, 64, two neurons, for instance? And I never manage well to understand that. But some convolutional neural networks comes with more layers and decrease the number of layers, and ended up at the end with two neurons classifying yes or no binary decisions. Okay, basically it's a very dense layer, and then it will go um, uh, several layers of hidden neurons, and then we'll end up with two neurons or four neurons classifying if a pixel belongs to a number or its background or it belongs to a certain number. So what we did here, in order to summarize, not with very mathematical base, but more with more plain by saying with plain words, we assign categorical data. What is that? This MNIST dataset comes with a trained dataset that actually has labels. For these numbers belong to 9. These numbers should be 8. These numbers should be 6. These numbers should be 1, and so on. And then we will train the convolutional neural network with that. And then with the other part of the data, we do not have labels at all. We'll try to guess what's going on here. Okay? So we will build this one. And we have a very simple convolutional neural network here. look how many number of parameters we need to train. The param number here, I will write down here. The number of parameters to train. So we need to train here 4,920 parameters, up to that. So it's very computationally intensive, okay? And depending on the complexity of the neural network, it will be worse. You will require supercomputer for more dedicated tasks, okay? And we have two dense layers of neurons here. Um, and we'll try to explain then what is a neuron at the end of this example. What is a neuron? What is a perceptron, etc., etc. A convolutional neural network, more or less. I will try my best. But the global picture, the overall picture here is to have a hidden layer of neurons, and that will process the data, and then two neurons, just two neurons or four, will classify and will give the last decision on what is a pixel or this number, etc., etc. They will classify, okay? Because at the very end, this is a classification problem. And then we have the layers here, dense one and dense two, and see how many parameters we need to train at the beginning. 400,920. 401, sorry, thousand. Then we diminish to the half of that, to 262,000. And then we ended up with 5,000 neurons that actually will do the last work. So what the convolutional neural network is trying to do here is to take all the pixels into account, all the shapes of the images into account, pixel by pixel, multiplying the pixels or doing data augmentation of the pixels. We'll take all these features, how the pixels behave in general, how the shapes of the number are, um, how the nines came in this data, or the number ones, if we can distinguish from 5 to 6, for instance, stuff like that. How hard it is to distinguish within numbers. All these parameters are trainable in a hidden manner, like in a black box. But what I like from this method, it's you do not need to parameterize so much. Okay, it's not the parameters are not so much complicated. It comes actually by default. Okay, uh, so the only thing you need to optimize here is the training step here. And then we will see the metrics of validation or accuracy. Okay, so we will do this training, and what I try to be as much as clear here. We will do the input data labels here, the number of images that are entering into the model in batch. It is one is little, you have to, this is the model, I will say here the model learns in batch. This is batch translation because I had my notes in Spanish, sorry about that. Okay, so this is the number of épocas in Spanish. So we will train this convolutional neural network in 10 epochs. We will do a validation split, which is a portion and training data, and you will see more or less how the model behaves with the data. Verbose is it will give you an output into the screen of the Jupyter Notebook, and we'll see what's going on here. Okay, so it's actually training with the data, MNIST data, 60,000 images at once. As you can see here, this model is very optimized to handle a lot of data at once. So my computer doesn't have any single problem here to do that. It's taking a while. And then we'll finally do the prediction and validation here, which is the loss of an accuracy. So we have an accuracy according to the model, the small model we trained, of 0.93, which is 93.4%. Okay? Do you think this is good or bad? What do you think? 93% of accuracy in general. I'd like to see what do you think about that. Bad, good. It's good but not good enough. Good. Actually, it's very good for the deep learning. Yeah, it's I did this specific example because it's very nice. Sometimes you get into 70%, but what if I tell you, for instance, you don't need to change so much this convolutional networks and stuff like that because this is pretty much established. But what if you need to, for instance, here, you have another layer. For instance, you can copy and paste, for instance, another layer. You can sequentially add more layers. You will increase in 100% accuracy. It will be perfect algorithm. Out of 10 will be wrong. Okay, yes, 1 out of 10 will be wrong. What if you have the perfect algorithm, but suddenly you change the data? You change the data entirely, and the algorithm performs very badly. What do you think about that? So, why this algorithm works 100% in a specific subset of data, and suddenly you change the data and works and behaves very bad? Why do you think this is happening? Do you have any thoughts? You can say it aloud. For because of the data used for training the model, yeah, overfitting. Adan, can you? Yes, it can be overtrained. Adan, can you explain in plain words what's going on here in overfitting? What is overfitting? Overfitting.
Um, okay, hello. Overfitting is when the model try to understand in something of the data that don't have has any relationship between the data because when more layers the model have they do in all of neurons linear regression. So the model tried to adjust to a line that there is a function of the regression linear. The linear regression is a linear function. So all the neurons do a linear regression individually. So when you have a large of neurons, okay, the linear regression of all neurons try to fit so much to the data that is more influenced by the how I say noise. That's the word. They
Yeah,
try to relate, try to find a relationship between the noise of the data. So that's not the solution. The model try to predict the data, but not predict the noise of the data. So we can put a lot of layers because the model will try to understand the noise.
perfect. Thank you very much, Adan. Actually, that's very true. The noise could be affecting the predictions as well. Something which is we thought is noise actually is not. Um, and that's very true. But the overtraining, the training, I can say, for instance, that the model try to learn by memory. Have you ever memorized a speech, for instance? I did all the time at the university. Uh, Polynomials of numbers of the yes, could be something like that, Francisco. Um, actually, I would like to explain it in the following manner. What if I memorize a speech and I have a sense that what I'm saying sounds very nice and very good and very classy? But what if someone asked me a different question that I was not prepared at all? I will fail because I just learned by memory. But I didn't comprehend the data. What's going on here with the hidden layers and the complexity of this data itself? I didn't take into account uh, other features. And you want your model to be able to generalize in the data, to say, okay, what if I have an accuracy of 60%? Very bad. But what if this model behaves very good regardless the type of the data? That's what I want to show. And for instance, in the terms of breast cancer or melanoma detection, the models will be behave at the 75%. But for instance, what if I'll tell you that you have 75% of this um, um, uh, patch on your skin will develop, this nevus on your skin will develop as melanoma. Will you go to see the doctor if I tell you 75% probability that this nevus will become to melanoma according to the machine learning? In my case, I will go, even with the 50%. Okay, so this is the aim for the a good convolutional network to generalize as much as it can. Uh, Basically, that's for instance just one example of um, how good a convolutional network could generalize within models, within data, and not just to learn your data and be very good at this data, but when you change the scenario, everything fell down. Okay, and the prediction. So this is just the NumPy predictions. It's not very important. And this is the matplotlib plot. So we'll see what's going on here. True is 9 here because we interrogated the label data, and we try to predict on the label data. Okay, this is the final step of the deep learning. The true is 9, but it predicts a 7. Maybe, I don't know, because a 7 is here. I don't know. True 9 and predicted number 2. This is very bad behavior, I believe. True 5 and predicted 1. Maybe it rotated the data and it saw a 1. I don't know. But actually, the deep learning rotate the image as well. So that's a very important feature of deep learning that actually is restricted in ordinary analysis. Deep learning can rotate and augment the data. True 1, prediction 4. That's very bad. 5 and 1, for instance. 6 and 4. 0 and 9. Yeah, because I can see what's happening here. Probably the model thought that the all right thought that actually it was a 9, but it's actually 0. 3 and 6, prediction 6. Can't see the 6 here. True 4, prediction 9. That's very bad. Maybe it thought something like that. 4, 0 and 6, 6. That's good. So next class, we will see what's going on with the deep learning with our own data. So I will teach you in more detail here because this is for image. I will explain step by step because I have the set stack loading. I will do some enhancement. We'll do a binary classification here. We'll label the regions We'll filter if the area is over 2000, which is the size of the cell. We will select certain nuclei to train. We'll do a training and validation accuracy, and we'll try to detect the nuclei here with deep learning. You can see here it actually fails, but with some parameterization, this number started to become better. And this is one you want to shift here. This one to divide because in the other example, uh, 3 cells were detected as one. Here you can see the cells were neglected in the previous example and actually are detected here. You can see this is a very bad behavior, but what if you have a set stack of 100 images and 100 set stacks? Good luck with that counting nuclei. Okay, so yeah, so it's we'll see that in the next class, and I will encourage to you to try to apply your data with some microscopy images if you have beforehand. Need to do a poll here. How many people here do have microscopy images ready to analyze? If you can write on the chat, will be helpful. Or say it. Francisco, Adolfo. I will have it for next week. Perfect. Imaging today. Perfect. Good luck, Tyler. I'm very excited to see yours. So maybe we can quantify intensity or something like that, or distinguish shapes or something like that, or localization. Yes. Okay. Can you please send me an email with set stack or something you have it, and I will prepare something very briefly on how to load your image on Python. And then you can, because once you have your image loaded on Python, then you can apply all these things, okay? With the aid of ChatGPT or Perplexity or Stack Overflow itself, or scikit-learn documentation if you want to go straight with the documentation, which is the best course of action, okay? But once, if you have all the data loaded into your environment on workspace, I will send you a piece of code to each one. So if you can send me through between today and tomorrow over the weekend, uh, it will be nice. So I can work in next week on that, and by Thursday I can load every single image for you. Okay. And then the immunofluorescence and access with imaging, genomics, and deep learning. And if you have fluorescence, if the Tyler, if they can give you a set stack or something with fluorescence, or the image in certain format, we can work on that. That's good. As long as they give you the raw data is good. So you can send me, okay. So yeah, you can send me that for training purposes, and we'll load it on the Python. And every single view, if you have something interesting to see, better images for you from that mine for sure, please let me know and send me one picture, and we'll see what we can do from there. And in the very last class, I would like you to present briefly 5 minutes to share screen what you have done. If you want to do a deep learning, what we can do with that, or if you can do puncta counting, we already covered that, or segmentation, and so on. So next class we will see how this deep learning behaved in terms of counting the nuclei and segmentation. Probably there are better tools, but we'll see how the deep learning goes. And then we will try to load your images and see what's going on Python. Okay. My guess is it will be fluorescence quantification. And probably shape this cell clustering as well. So okay, so this is the end of the class. I hope you guys enjoy. I know it's kind of dry topic, deep learning, but it's very useful. So if you can search a little bit more about the parameters and what we did here, is good. But this is just an example, or you don't have to learn it, but it's an overall example of what's going on with the deep learning. So next class we'll get into more details. I will do a previous class of deep learning in order to understand the concepts, and then we'll go into the nuclear segmentation. But the problem is how to distinguish the true number of nuclei in an image. Okay? Well, thank you very much, guys, and I hope I can upload the class for next week. See you Thursday. Thank you very much, guys. Take care and have a good weekend.
Bye.
Carlos.